Spatial Orientation by Allison Arai, Katherine Chao, and Amy Streets. Spatial orientation is how animals figure out where they are in their environments, and most spatial orientation experiments study rats. This is Daphne the rat, the star of our show. And this is Daphne's brain. Place cells are located in the hippocampus. Each place cell has its own place field, or a specific location where it fires. As a whole, place cells represent each location in an environment and function to tell an animal its position in space. If we record from this place cell, shown in blue, we see that its place field is in the upper left-hand corner of this environment. So as Daphne moves around the arena, the place cell only fires when she is in the upper left-hand corner of the arena. Grid cells are located in the entorhinal cortex. Grid cells have multiple firing fields that extend across an environment. Each grid cell has a different distance between firing fields. This creates scalar representations of relative distances in the environment. Different firing fields from different grid cells overlap to help an animal determine its location. Recording from this purple grid cell shows its distinct pattern of firing fields in this environment. This second grid cell has smaller distances between its firing. Recording from this dark blue grid cell shows that it has twice the number of firing fields as the purple grid cell in the same amount of space. This is the subiculum, home to head direction cells. Head direction cells act as a compass and fire only when an animal's head turns toward a specific direction. Recording from this red head direction cell shows that it fires only when Daphne's head turns about 45 degrees away from center. Daphne can move to other locations, but the head direction cell will only fire when her head turns 45 degrees away from center. Border cells also reside in the subiculum. Border cells describe an animal's distance away from and direction relative to a boundary. Recording from this border cell shows that it fires when Daphne is on the south side of a boundary, whether that is the wall of the arena or the side of a blockade. Grid cells, head direction cells, and border cells all send information to the place cells. Place cells also send feedback to grid cells. Thus, the place cells integrate sensory cues and other directional information to determine self-location.
How does a place cell in this first environment, a forest marked by this tree, fire in a second environment, a meadow marked by this flower? The spatial organization system remaps whenever a new environment is encountered. Thus, a place cell that fires in the upper left-hand corner of the forest might fire in the middle right area of the meadow. Cells also, remap upon encountering new environments. The dark blue grid cells in the forest have large spaces, whereas the dark blue grid cells in the meadow have smaller spaces, producing a smaller grid. Cells do not remap when placed in new environments. Thus, Daphne's red head direction cell responds only when Daphne's head turns 45 degrees away from center. Border cells also do not remap, so Daphne's green border cell responds when she faces the south side of borders.
So what exactly does this mean for Daphne? It means that her hippocampus not only stores memories, but can also predict the future. In an experiment, researchers trained rats to take a certain path towards a reward. The researchers then blocked this path and tested whether the rats would know to take a new, alternate path, or if they would stick to their learned path. This is the learned path demonstrated in a black dashed line. They found that the rats had no trouble finding the alternate path, even though the rats had never learned it or been taught to follow it. Yay! Cheese! Recordings from place cells demonstrated that the rat cells knew which way to go well before the rat moved and did not require any investigation of the barrier, shown here in red. This simple experiment shows that the hippocampus can both remember past locations and paths, as well as predict new paths when the environment is altered. Here is the rat following the alternate path in red.